And a very warm welcome to you, and thank you very much for coming on this chilly Edinburgh night to this wonderful lecture theatre. And I'd also like to extend a warm welcome to our friends who are hopefully out there in cyberspace listening to this streamed lecture. Surely one of the most interesting and deep and compelling questions in science is to ask, how is it that we developed this complex set of behaviours, not only in humans, allowing us to do many complicated components of our psychology, allowing us to contend with complex situations such as this microphone here. Fine. Is that better?
Yeah. It's not screaming.
Thank you. And I'm sure you can hear me because it's a wonderful lecture theatre. So one of the great questions in neuroscience is to ask, how did our wonderful complex set of psychological functions ever arise to enable us to do all of those sophisticated things that we do on a daily level? And it's perhaps through having some insight into that that we might be able to understand the flip side of it, which is to ask, how is it that the diseases of the nervous system, which can afflict children and young adults and elderly people, how is it that they come about? And what is it about the mechanisms of the brain which conduct our normal behavior? What is it about those mechanisms that ultimately goes wrong? One way to begin to think about those problems is to ask this really quite simple but fascinating question, which is, how did intellect and disability actually arise? And that's going to be the focus of my lecture this evening. And to help you think about this, and when intelligence, feelings, and emotions, and other components of psychology might have arisen, I'd like to show you a very simple tree of life, which just illustrates just several species and shows you some of the relationships between them. Here are some vertebrate species here. And you've all heard that humans and chimps have about 96% of DNA the same, and that's because they had a common ancestor about 6 million years ago. And these primates had common ancestor with rodents about 75 million years ago, and so on, and back to simple invertebrates, and earlier to single unicellular organisms. Now, one of the characteristics that separates these different types of animals is that we all know that us, and in fact other mammals and other vertebrates, have wonderfully complex behaviors, whereas invertebrates have simple behaviors. How is it that we obtain this much more complex behavior, something that is sometimes referred to as the cognitive Big Bang, the expansion in the way that we can think and reason? How did that actually ever come about? But I'd like to think about that question and set out for you the very broad history of life as we know it. And this is a timeline extending from the present day on the right all the way to about 4 to 5,000 million years, 4 to 5 billion years ago, which is when the Earth first formed. And we of course know that humans have been on the planet for the last 5 to 6 million years. And it was about 500 million years ago that we first find in the fossil record the first evidence of multicellular animals, various kinds of invertebrates and then vertebrates. But it was much earlier than that life is thought to have begun on planet Earth, with DNA forming in rock pools in this violent world that lived there, and the very first cells were formed about 3.5 billion years ago. And in fact, you can find in the fossil record, just over to about 3 billion years ago, the first evidence of life in the form of bacteria. But there were cataclysmic events going on the planet. For example, there was at one point an ice age like no other, when the planet was covered with a kilometre thick of ice over the whole planet. And after that thawed, we saw some of the first more complicated single-celled animals known as eukaryotes. And about 700 million years ago, there was yet another snowball Earth covered by ice. And it was after that thawing event that we first start to see in the fossil record the first multicellular animals, which arose about 500 million years ago. Now, what is it about that and the brain? Let me first introduce you now to some very simple classical view of the nervous system, and from that we'll begin to explore these evolutionary mechanisms. The classical view of the brain that I will now present to you is the sort of view that arose in the late 19th century and earlier 20th century, and it essentially revolved around the idea that the brain is made up of many different cells and there are electrical reflexes in the nerve cells of the nervous system. And this just gives you some idea about how sensation from the external environment, in this case a sharp pinprick on the finger, will activate receptors which will then engage and drive action potentials, the electrical activity that goes down the nerve fibers, ultimately traversing those nerves and finding its way back into the spinal column, and from there going up into the spinal column, into the central nervous system, or into the brain. Now, within the brain, there are special contacts between the many nerve cells, and this is a picture of one of them here. And this is, of course, the synapse, the junction between nerve cells. And the electrical activity, as it comes to this terminus of the nerve cell, can no longer continue across this gap, and the only way that information can be communicated across that gap is because synapses have the capacity to release chemicals which are stored within these little balloons containing those chemicals, the neurotransmitters. And when the electrical activity comes down, those small balloons are induced to fuse with the membrane, releasing their contents, and those chemicals, those neurotransmitters, diffuse onto the synapse on the other side, on the other neuron over here, where you can see there are these neurotransmitter receptors in the surface here. When they become activated, they also now trigger off an electrical activity which then further propagates through the nervous system. And it was this idea that nervous impulses come into the nervous system and get trafficked around through these dendrites, through these synapses, which was really at the foundations of electrophysiology at the beginning of the 20th century. And we owe an enormous debt to the pioneer of electrophysiology and the reflex, Charles Sherrington. Not only do we owe him a debt for describing how this sort of activity works, but also because he coined the word synapse from the Greek word to clasp, the junction between nerve cells. At the same time, or perhaps a little earlier, was another seminal insight into the organization of the nervous system, and that was brought about by the microscopists. And Santiago Ramon y Cajal is probably the finest example of anybody who looked down a microscope into the sections of brain tissue. And here is a picture of him in his laboratory and with his microscopes here in Madrid. And he was really the great pioneer. And here's one of his original specimens. And what you'll see in that specimen is the fact that nerve cells are peculiar because they have these extraordinary tree-like branches on them, like no other kind of cell. And he surveyed these in many different brains and organisms, and some of his famous pictures are shown here, where you can see these tree-like branches, and on the ends of these branches, where the leaves of these trees might be, that's where you find synapses. And in fact, there are many cells, there are hundreds, if not thousands of synapses on a single cell. And we know in the human brain that there is an order of 1 billion synapses in the human brain. And it was this sort of anatomical perspective that one could ask, as they did, about the origin of the brain from the anatomist's view. And it was really quite simple. If you can find nerve cells in organisms, then that presumably indicates where the brain might have evolved. And it was well known that jellyfish, which are thought to be one of the earliest types of multicellular animals around, which has contraction and can move and can swim, and indeed is in the fossil record, and you can find them in the order of about 600 million years ago. It was thought that this was the origin of the nervous system because it had such a simple nervous system in these animals. It was in fact around about this time that there were many other organisms that appeared in the fossil record in the Cambrian period. There was an explosion of diversity, initially of the invertebrates, of which you can see many examples here. And it's quite obvious that they had complex eyes and complex movement, social interaction. So they clearly had nervous systems and had many characteristics of their descendants who live on the planet today. So with that sort of perspective, the anatomists could see the idea that the brain originated about 500 million years ago with the origins of these complex and beautiful animals. But what did they have to say about the cognitive Big Bang, the expansion in complexity of behaviour that occurred after here? Well, the anatomists thought that the nerve cells were so important and the electrical activity was so important in those nerve cells that probably the most likely explanation is that these organisms developed much larger nervous systems. And of course we know animals of various kinds, and their brains are shown here, have these very large nervous systems, these vertebrates do, and that presumably is the explanation for their much higher capacity to perform complex forms of behaviour. But is that true? And I'm afraid to say that there is no scientist today who can actually tell us that it is, because we simply do not know. It's never been actually tested. But now I'd like to turn to some of the newer insights, in this case in the 21st century. This is an exciting era because it is the era of genomes and proteomes, where scientists now have the capacity to find every gene and every protein that works in the brain or indeed any organism. So now, with these sorts of tools, what might we learn by looking at the synapse? And a simple question to ask is this: how many proteins does one find in the synapse? And as recently as 1990, it was thought that there was probably a few dozen proteins that you can find in the synapse, and that's all you need to perform this activity of transmitting the information. But it turns out, 20 years later, that we know that there's more than 1,000 different proteins in the synapses that you'll find in a human brain or in a mouse brain. It's remarkably complex. And why should it, and would it be so complex when you apparently need so few proteins to conduct the job of the reflex arc? We not only know how many proteins there are, we actually know the parts list. We know the types of proteins. And we also can begin to understand how the different proteins might fit together, because it is proteins that can actually assemble together in the synapse. And here's the membrane of the cell, and here's a neurotransmitter receptor. It doesn't just sit there all by itself. It binds on to other proteins, which then tether other molecules together and can form little multi-protein machines. And these little machines do the work that's inside the cells, and they can have remarkable properties. For example, these sorts of molecular machines that attach to the receptors and other proteins here can act as computers. They can detect signals from the neurotransmitter and work out the patterns of activity. And this slide here is of a molecular circuit diagram of the proteins inside a mammalian system, a synapse. And here's a lot of different proteins connected up. And you can think of this as being like a computer chip, where there are many different components, hundreds, if not thousands, if not millions of transistors and components that within one chip are capable of enormous extent of computation. And we now know that the molecular components inside synapses are capable of an extraordinary amount of molecular computation, perhaps being able to make billions of computations within an individual synapse. So this remarkable power and sophistication at the molecular level that's being exposed by this complexity only makes it more compelling to ask, how is it that this ever came about, and where did it in fact come from? It also allows us to think again about how complex or how powerful the brain may be when one thinks of it as a computational device. And people have revisited this question and thought about how much computational power this molecular machinery confers on the nervous system. And it is thought now that the human brain may in fact have greater computational power than all computers that currently exist on the planet. But now let's look into some of the molecular machinery within the synapses here. And I just want to remind you that genes make RNA, make proteins, and they make synapses. And of course, I've just shown you some of this wonderful molecular machinery in the synapses here. And here is DNA at the bottom, and it makes the RNA. It makes individual protein components, and they're assembled into those machines, and they're built into the synapses. So the beauty of this is that scientists now have not only the capacity to examine all of these different proteins and all of these different parts, but we actually know the DNA code from genome sequencing of all of those different parts and in many different organisms. And this really opens up the door for some exciting studies on the evolution of synapses, We can ask, how did those parts ever evolve? Where did we first discover synapse genes? What sort of organisms have them? Which ones have the same ones as humans? Which ones have the same ones as invertebrates or jellyfish? And we can now address the question of the origin of the brain, but from the molecular standpoint. Before I tell you some of the findings here, you need to understand, and I'm sure you do, something that is really, truly a fundamental yet simple way of thinking, and it is what we'd call trees of descent. And there is no one more famous than the former medical student of Edinburgh University, Charles Darwin, in this context, because after he left Edinburgh, and you can see in his notebook here from 1837, this is possibly one of the most important scientific diagrams ever written, and it is to show his view of how different animal species may be related to one another by the similarity of their physical characteristics, and that one may infer descent from a common ancestor by the conservation of similar characteristics. And there were many other trees that were inspired by Darwin's work. Here's one from Haeckel, and I've also shown you this nice simple one here. And as I've alluded to now, because we have the DNA sequence, we can compare the DNA sequence of different organisms and from that deduce and triangulate back in time when those genes evolved that are in humans and in other species, and work out how the synapse genes evolved and when. Now, we don't just need to look at a few species like that, but because of the genome revolution, we have a tree of life that looks something like this, where here are the names of individual species around the perimeter of this circle, and you can see the trees of branches here indicating the relationships of these various species. And in fact, there have been fully sequenced genomes for thousands of different organisms today. And I'm now going to show you what we've been able to learn by examining the molecular origins of synapse proteins. Here we have those sets of proteins that one can find in the synapses of humans. And you'll find that, remarkably, that all of these other vertebrates have more or less the same set of proteins that are there. But if you look into invertebrates, you'll find that they have, again, the same sorts of proteins, but you'll notice there's fewer of them. It's the same type, but there are fewer. And quite remarkably, particularly if you thought that the brain was doing very sophisticated and complicated things, creating thought and reasoning and responses to the external world and creating language, then you might say, well, of course the synapse is going to have all these wonderful magic molecules that do all of this kind of thing. But it surprisingly turns out that the same molecular machinery exists in animals, unicellular eukaryotes, that actually have no nerve cells at all, of course. They're single-cell animals. And the fundamental molecular machinery, these molecular computers, can actually be found in those organisms as well. And moreover, back in bacteria, there are even some of the components that you still find in the human vertebrate synapse. I'm going to show you more about that and show you a few of the depths of the origins of synapses and the deep ancestral history that that has. Let's now look in these single-celled organisms, and here's one of our single-celled eukaryotic organisms swimming around in its environment. Its DNA inside its nucleus here, and these sorts of organisms lived 1.5 billion years ago on the planet, obviously about 1 billion years before any multicellular organisms, and you can find in those organisms the same sets of proteins which we refer to as the protosynapse, the ancestor of synapse, that set of proteins that you will find in your synapses or indeed other vertebrate species. And what's more, in the same way that those proteins are controlling the way you detect and you respond to your environment, they are doing the same job in these unicellular eukaryotes. So these synapse proteins are used for sensing their environment. Now, I find this really quite striking, and it is when you look at this tree of life here, and you look at the very earliest points, what is called the universal common ancestor of all living things, that you will find that there are synapses and the brain began near the origin of life, and that is because you can find in these prokaryotes or bacteria sensing proteins on the surface and their signaling proteins within the cell. The very same sets of proteins exist and work in synapses of humans and other vertebrates. Those machinery have been used in sensing for the last 3 to 4 billion years. It's quite fascinating to ask, how and why would those have come about? They're obviously very, very important. And if you think now about what may have existed before cellular life, and the scientists who study this are quite fascinated by how DNA and RNA may have formed in these ancient rock pools under simple chemical reactions, but somehow they couldn't really expand and grow and become very complicated forms of DNA unless they were somehow protected from their environment. And at some point it's thought that there was the first cell, it's called a protocell, where this membrane wrapped around the DNA and packaged it and protected it from the external world. Now, for this type of protocell to become more sophisticated, presumably there was the evolution of these receptor molecules of the kind I've just mentioned for you, which would allow these very simple early bacteria to detect and respond both nutrients but also toxins and move and swim and move away from those. So these simple sensing machinery was probably a very fundamental and basic form of behavior that allowed these organisms to protect their DNA and thrive and flourish. So with this sort of history from the molecular point of view, we'd now like to revisit this and say that the anatomists could only look back about 500 million years, but the molecular scientists can now take us back way into the distant past and show that the origins of the nervous system, or key components of the nervous system in synapses, the origins of synapses, brains, and behavior, exist right at the very earliest origins of animal life. Now, don't get me wrong, I'm not suggesting that the 19th century scientists who were extraordinary in their capacity to study animals in the natural world didn't have profound insights or views on these kinds of matters, and I rather like this quote from Lloyd Morgan in his book Animal Life and Intelligence, published in 1891. Because what he says is that the object of the receptions of the stimuli of the external world or environment is to enable the organisms to answer or respond to these special modes of influence. Now, in the unicellular organisms, where both the reception and the response are affected by one and the same cell, the activities are for the most part simple. Though even among these protozoa, there are some which show no little complexity of response. So, we have a deep ancestry of the synapse in this unicellular world, in these basic forms of behavior. But now I'd like to turn our attention back to the vertebrates and the higher vertebrates. And in particular, I would like to address this issue about how it is that there was what we call the expansion in complexity of the synapse to become from a synapse in the invertebrates, which had this capacity to do all of this signaling, and why is it that there's so many more of these molecules that you find in the various vertebrate species? And I think this is really a very useful way to think about it. Here you have this machinery that's capable of doing all sorts of wonderful things in these unicellular organisms. And it became a little bit more complicated when the first multicellular organisms came about. But for some inexplicable reason, we have this. Now, why, you might ask, would we want to have that? And the question I'm going to put to you, or the hypothesis, is to ask, did this complex synapses actually help build complex behaviors, all of these complex behaviors here? And how was it that somehow during this sort of Cambrian period when there were these various invertebrates running around, how is it that we went from them and their sort of relatively simple behaviors and move up towards those much more complex behaviors that are characteristic of the vertebrates? And I think the answer is to be found in this picture here on the left. And in this picture here is a small animal for which you will find a fossil in typically the sort of dustiest corners of the museum. And there is a very fine one sitting in the Museum of Natural History in Paris. And it is a picture of this wonderful little creature here, which is the ancestor of all vertebrates, and it's something called Pichia. Now, let's explain how it was that these invertebrates and Pichia parted ways so that Pichia's descendants could become those animals which we know of as the families of vertebrates. And it was around about 530, 550 million years ago, with the complexity of life, that there was the evolution of the vertebrate jawless fish, various other kinds of fish, and eventually animals crawled out onto the surface of the planet around 380 million years ago, amphibia, then reptiles and birds, and of course various mammals. Now something very special has happened because they have got this remarkable complexity of morphology and diversity and capability to occupy many different components of the environment. So something really very special has happened down here at the root of all of these vertebrates. And I'm now going to introduce you to something quite remarkable. Of course, you will have heard of mutations in DNA, how the sequence of DNA can change maybe just by an individual nucleotide or a base pair, and that can have a profound consequence on the organism. But now I'm going to introduce you to the biggest mutation of them all. And this is the type of mutation that didn't just change a single nucleotide, it changed the entire genome, and that there were organisms that had an entire extra copy of the genome that arose. I should point out for you that this is a remarkably rare event, and it has been described several times in plants. It has been described, the last time it has ever been described in the evolution to humans was about 550 million years ago, the events that I'm about to tell you about here. It was a remarkable and rare type of event. Let me show you what it looks like. So here is a picture of the genome, the DNA of our hypothetical ancestor, that little Pacaya-like organism, and it was happily having offspring. And you'll notice here that's a perfectly normal offspring, but for some odd reason a genetic accident occurred such that one of its offspring, only one, had an entire extra copy of the genome there. And it survived. And oddly enough, not too much later, after that one copy, after that one event produced that extra copy, one of its descendants had an additional duplication event, multiplying effectively 4 times the number of genomes that its ancestor had. And this is a remarkable event because it was that single organism, and it's extraordinary to think that it was a single animal that gave rise to all of the vertebrates that we know today. You might ask yourself, whatever happened to its siblings? These other guys here. Well, I'll show you a picture of them. There they are. They're alive and well today. The lancelet, or Amphioxus, and there is a Pacaya fossil over there, a very rare and wonderful fossil. So clearly there was something very advantageous to having those genomes, and now I'm going to ask this simple question: what good are these extra genomes? And I'm going to explain to you why this was a really extraordinarily important event for the evolution of vertebrates and in the synapse and ultimately in the behaviour. But I'd like you to begin to grasp the idea that having these extra genomes in this ancestral animal was really the starting point for the evolution of new genes. Now, I know that you will know this, but there are 26 letters in the English alphabet, but only 4 letters in DNA. And here, of course, is some DNA sequence, and you can see the various letters printed here. And of course, here is some words that you can quite easily read. And I'll use the English language simply because it's an easy way to illustrate the principles of how mutations change various letters, either in DNA or in the English language. So we can think of the genome as the book of DNA that defines that species, and one sentence within that book we might think of as a gene. And the human gene, for your information, genome, has about 20,000 genes and about 3 billion letters of DNA. Incidentally, there's about 100,000 words in a typical Harry Potter novel, to give you some kind of comparison. Now let's try to understand mutation. I'd like to introduce for you mutation, and you will recognize how remarkable it is as the engine or the driving force of variation. It is the engine and driving force for evolution. And here's now a very simple organism. I'm just drawing it like this, Imagine this is a simple organism, and there is its DNA, and there is the sequence of DNA, and we know that sequence is functional because we can read it. It says, the quick brown fox jumped over the lazy dog. And it's a viable organism. But now we're going to obtain a mutation. So look at the DNA sequence, and here is our mutation. We've changed just one letter. The quick brown fox jumped over the lazy dog. Now that doesn't make any sense at all. And that animal will die. There is no longer the meaning or the function within that sequence of DNA. Now that means that if you have that sequence of DNA, you're very susceptible to the impact of that mutation. But now let's ask ourselves, what happens to mutation in animals that have this extra genome that I've just told you about? And here it is. Here's our animal. It has these two copies. This ancient organism got these extra copies of the genome. I'm just showing you two for simplicity. And now let's look at what happens with a mutation. And here we have it. The quick brown fox jumped over the lazy dog. No problem, because the other genome is there, and that genome, that other genome being present, allows that mutation to be tolerated. So this mutation is tolerated because we have a spare or backup genome over there. And so that is very useful. But what is also rather extraordinary and useful is that over many years, new mutations can actually accumulate. So now let's have a look at this sentence here and see how it mutates with time. And every so often, a new mutation will turn up, and as you can see, the letters of that other sequence eventually are changing. And with those changes, which typically occur over many millions of years, possibly as much as 50 or 100 million years since the time of the duplication event, what can happen is that this sentence changes meaning altogether. So rather than the quick brown fox jumped over the lazy dog, the quite urgent fox helped save the boy's dog. Now, this is rather simple and straightforward, but I hope you'll understand that it is through that duplication and then the subsequent diversification through accumulated mutations that having those extra genomes now produces extra genes. And we know that to be the event that made these simple molecular machines that were in the synapse of the invertebrates become ever so much more complicated in the vertebrates. And in fact, for many of these vertebrate genes, there are 4 of them that are present in the genome that all derived from that ancestral individual one of those. So this genome duplication event was an extraordinarily important force for generating a molecular toolbox that expanded the complexity and diversity of all of the different vertebrates and in different parts of their body systems. And the question we would like to ask is, did that matter in terms of the evolution of these complex forms of behavior? Well, I'm going to show you an experiment now, and I would like to introduce you to something that psychologists like to get their teeth into, which is the complexity of mammalian psychology and human psychology, and I'm describing it here as the repertoire, the set of behaviors, the sets of forms, of complex forms of thinking, learning, memory, and attention that mammals, including humans, have. There are simple forms of learning, complex forms of learning and executive function, and you can see they're grouped and classified here, and I'm not even going to try to explain those for you today, except to let you know that psychologists measure these things, and they measure them with all kinds of different tools and tests and tasks, with paper tasks, with computers and so forth, and humans can do these tasks. But something that you may not know is that not only can humans do these tasks, but so can other animals. And in fact, mice can do these tasks, and you can get mice to do these tasks by placing them in front of something essentially that is an iPad. It's like a tablet computer. They will touch the screen when presented with these images on the screen. They will wander up and touch the screen. I'll show you how they do that in just a moment. And you can ask them to do all sorts of tricky and complicated things, measuring these components of psychology. You can do simple forms of learning, very complicated forms of learning, and measure flexibility and ability to change their mind and various types of attention. And you can measure these tasks in mice. I suppose you'd like to see how it works. Let's take a look. Right. So on the left, you obviously have a computer screen, and the image will come up. The mouse is having a little snack. Oh, there they go. There we are, it hit the correct one. It gets a little reward, a little snack again. No pain or suffering, it's just getting a reward for its work. Images come up, and it picks the correct image yet again. Those of you who think rodents, and indeed many other species of lower animals, might not be very clever and sophisticated, will be shocked and surprised to see how clever they can be when confronted with these sorts of tasks. Now, given that, let's now do the test. We're going to ask the question, did those new genes that evolved, could those new genes have influenced and shaped or made those behaviors more complex in their regulation? And so how do we do that? I've told you that there was an ancestral duplication event that multiplied the number of genes, and we're going to take one gene family, which in invertebrates, there's only a single gene, and the invertebrates that live today only have a single one of those genes. But because of these duplication events, there are 4 of them, and in fact in mice there are 4 of these genes. They have this lovely little acronym called DLG. We won't worry ourselves with what that actually means. But we have mice, and here's the important thing, we have mice that have been genetically modified so that this mouse here does not have the DLG1 gene, and this one does not have the DLG2 gene, and so on and so forth. And so we can take those mice and we can ask them how well do they do these certain tests and tasks over here. And what we're looking for is to ask, if those genes have really diversified, then they will show different effects in those different aspects of behavior. And that's exactly what we see. So here we have our set of different behavioral tasks. These little boxes represent the results of the behavioral testing. And here's our 4 different genes, 1, 4, 2, and 3. They're all listed across here. And the only thing I really want you to see is that the colors, that is to show that the genes are having effects, are shown by the colors, that the colors are different. In different parts of the behavior, different genes are doing different things. And what's rather remarkable, and something I'd like to talk about, is that for certain behaviors, and I'll pick on this one here, you'll see this gene is showing a red response, and this one is showing a green response. So they're actually doing the opposite kinds of roles of regulation. So this means these ancient genes have diversified to control and amplify and attenuate different aspects of behavior. Let me show you just one example of how that works. Let's ask them to do something a little bit tricky, which is flexible thinking. Not just the ability to learn something, but the ability to now unlearn and switch to something else. Flexible thinking. So a mouse can be trained to respond to this little image on the screen, and you give it a little reward, much as you would if you were to come into work and be paid every week. But then what happens? You no longer get paid. He no longer gets rewarded. You still come in, at least the next week you come in, and maybe the next week. And after a while, you sort of gradually lose interest. So if I was graphing that against the number of weeks, it would actually go down with time. That is showing that the animal has now beginning to change their strategy. They're beginning to be flexible. They don't just continue to stay in here. They begin to be flexible. And we can see now what happens in a mouse that lacks that gene called DLG2 and lacks that gene called DLG3. These mice are less flexible. They're slower. These ones are more flexible. They're actually faster. So that just shows you how those genes are controlling this cognitive flexibility, the ability to change their thinking. Now, this is thought to be really very important because it is essential that when animals are in complex environments and changing situations, that they need to be flexible to survive. And here's a nice example which I just recently saw on the BBC website on an island just north of Alaska called Cooper Island. And on that island recently, guillemots have been breeding on this island, but as over the last 2 decades, the sea ice has been melting more and more, and they have moved further and further out to sea. They've had to change the way they feed and how and where they nest, depending on the distance to the food. And it's this kind of cognitive flexibility without which animals such as this might readily become extinct. And having this flexibility, which has occurred through vertebrate gene evolution, is something that is no doubt very important and for those animals to succeed. And success in such environment depends on versatile response control and flexibility, which is something that has come about through the evolution of those synapse genes. So with that type of experiment and many others like it, we've seen very strong evidence that these ancestral genome duplication events that multiplied these genes, creating this complexity of synapse proteins, has contributed to the complex behavior that is characteristic of the vertebrate species. And therefore, it is this key event in genome evolution and synapse evolution that has contributed to this wonderful diversity of vertebrate behavior. It's very interesting to think about this from the point of view of mammalian evolution. And here's another timeline where I'm showing you the present day, going back about 100 million years. And you'll notice this little creature off to the left over here. That's a tree shrew, thought to be the animal that was the ancestor of all mammals. And had they been around when they were filming Jurassic Park, it would have been wandering about as the dinosaurs were tramping around. And it was about 60 million years ago when the big Chicxulub impact occurred and finished off all of the dinosaurs, rather thankfully, I would say, because possibly without which these other mammals wouldn't have ultimately evolved into humans down this particular lineage here. But you'll see that there are many different mammals that have arisen in addition to humans and to mice. But of course, you'll be asking yourself the question, what about humans? You've been telling me about vertebrates and mice. That's all very interesting. But you'd like to ask the question, are those genes doing the same thing in humans? But surely not, because after all, mice have a little tiny brain, and humans' brains have expanded 1,000 times bigger than those of mice over the last millions and millions of years. And surely those genes are not going to be doing the same thing. Well, we might actually be able to look at that question because, as I've already told you, that mice doing the iPad is one thing, but humans doing it is just the same thing. And so we would very much like to know, do humans and mice use the same genes for complex forms of thinking? How might we do that? Well, I've already shown you that it's possible to measure forms of complex behavior, rule acquisition, visual spatial learning and attention and so on in the mouse. And I've shown you, and here's just a scientific way of measuring these complex behaviors here. And here's this DLG2 gene, which I've already just very briefly mentioned to you. And this is the sort of measurements in a mouse that is lacking that gene. These are the sort of the abnormalities that that mouse shows in those different complex forms of behavior. And what we've been able to do is to study the role of that gene in humans. And it is because some of our colleagues in Edinburgh have identified humans who carry mutations in that gene, rare mutations that do occur, and those people have had the opportunity to be tested on the same types of touchscreen tasks that one has used on the mice. And therefore, we can measure the same psychological functions on a human on one hand and on a mouse on the other, and both of them are carrying a mutation in the same gene. And we can ask, do those genes do the same thing? And there's the answer. They do. And here is the performance in the humans, and the same sorts of impairments are seen in both mice and humans. This has many consequences. One of them is to just now go back to the idea of the tree of descent and common ancestor. It tells you that that gene was controlling those four aspects of cognition, those complicated forms of thinking, a long time ago in the ancestor of mice and the ancestor of humans. It also tells you that those functions have been maintained during that 100 million years, despite the enormous growth in the brain of humans. So it is quite clear that mice and humans not only share similarities in their behaviors and their mental functions, but more importantly, they also share, as you can see in these experiments, underlying genetic and synaptic mechanisms. Now, of course, our famous former medical student has written extensively on the subject of the similarities in behavior of vertebrates, and no other place you'll read this better than in The Descent of Man, published in 1871. And in this book, and many others like it since, there has been an enormous amount written about the similarities of different vertebrate species, in particular mammals, but also birds, with respect to their complex forms of behavior. And we're now beginning to uncover their common genetic and molecular underpinnings that have originated in those genome duplication events. Many of you, of course, will be fully aware that there's more than 20 different species that exhibit complex use of tools and mechanical tests and tasks. We know nothing about the genes involved with any of those, but it's very likely that those mechanisms that you might find that control tool use in birds or in rodents or in fish even where there is limited tool use, and also, of course, in primates, may in fact be using the same sets of genes. Now, let's return to humans, and let's ask ourselves a little bit more about what those genes are doing in humans. I've shown you that touchscreen test, but it's not just a case of humans using those genes to perform psychological tests in the lab. People are using those genes to get on with their daily lives, and unfortunately, some of those same genes that have multiplied through this evolutionary event can become mutated in humans, as I've already alluded to, and they are really very important. And in fact, some mutations in these so-called new vertebrate genes, the genes that the invertebrates didn't have, can, when mutated in humans, result in disease. For example, I've mentioned this DLG2 gene, that individuals with that mutation have been reported to have schizophrenia. Humans with DLG3 gene have a particular form of intellectual disability, and that's just two examples. And I just want to return to this evolutionary question, because it's often said that for some reason or another, psychiatric illnesses are something that only humans have. But what this implies is, because I've already shown you that the function of this gene, which is linked to schizophrenia, is in both humans and mice, it's very likely that these animals are also suffering and impaired by those types of mutations if and when they do occur. And in thinking about this, I came across this delightful little note on a paper published in what wasn't, which was a sort of the forerunner of the British Journal of Psychiatry, in the same year that Darwin published The Descent of Man. And in this paper entitled Madness in Animals, Dr. Laura Lindsay, who's a physician to the Murray Royal Institution for the Insane in Perth, said, "While the said term madness has been borrowed by veterinarians from human medicine, it is evidently regarded in a different signification when applied to other animals than man. So much so that while the character of the mental phenomena in certain diseases of the lower animals must be, and is, admitted by all veterinarians, they deny nevertheless their parallelism to those of insanity in man on the ground that animals have not sound minds and cannot, therefore, possess unsound ones." If you read a little bit further into this manuscript, he goes on. He says, "Such a belief merely illustrates the utter ignorance and prejudice regarding the mental constitution of animals which appear to be characteristic of veterinarians as a class." I couldn't imagine writing that in a paper today. But then he has an asterisk with a footnote, and he goes on, and he says, "Dr. Cobbold has lately shown how utterly untrustworthy are veterinarians and butchers in matters of passing daily under their notice, in those relating, for instance, to the entozoic diseases of meat." I don't even want to know what those diseases are. But let's talk about madness and mental illness and neurodegeneration and intellectual disability and other types of diseases that are known as brain diseases. And of course, you'll be well aware of those, and if anybody amongst this audience has had the, I'm sad to say, misfortune of having a family member involved, you will appreciate very much the social cost of these brain diseases. But I'd like to point out for you that the personal suffering of the individuals, as well as the social cost, is of course very important. But there is the remarkable economic cost of these disorders, and you can read across that slide if you wish. Now, what is it that we might learn by looking at the synapse and looking at these sorts of diseases? And I would just like to let you know that through the efforts of the research team who've analysed synapses from the human brain, we've been very interested in finding which diseases impact upon this myriad and cornucopia of proteins that you find in the synapses of the humans. And by examining those proteins, and again using the DNA sequence that encodes those proteins, we've been able to find that it's quite remarkable that there is over 130 brain diseases that are caused by mutations in more than 200 different genes that impact on those proteins. And just about every week in the scientific literature, more mutations are being discovered in these genes that encode those synaptic proteins. And it's clear that these are extremely important. That is to say, when those genes go wrong through mutation, that diseases arise. And there are many different types of diseases, and here's just a list, and I won't go into it, but those of you who are interested in these things might like to recognize the names of some of the many different types of disorders, which also include common disorders like schizophrenia, autism, intellectual disability, and dementia. And again, I want to return back to the origin of the genes that are impaired here, because, as I've already told you, these genome duplication events produce many different genes, but it is many of these novel genes, including those two I've already told you about, and many other disease genes that occurred in the vertebrate lineage, those are the genes that are impaired in diseases. So not only are those genes important for normal mental function, as I've described for you, but they're also important for disease. And this has led us to the thinking that it really is that this complexity and sophistication of behavior that arose in vertebrates, it comes at the price of susceptibility to the brain diseases. I'd like to say more about these mutations and show you a new insight that's come about recently, again by genome sequencing of families, where the sequence of the parents have been analyzed and compared with the children. And it's quite remarkable now that through whole genome sequencing, it has been discovered that every child has new mutations. Many of them don't matter at all. They're not inherited from mum and dad. They just turn up in the very earliest formation of the embryo. And sometimes these new mutations damage the synapse. So if one looks at the DNA of a mum or a dad, and they might have several children, occasionally there is a mutation that damages the DNA in one of the offspring, and that can land onto these synaptic genes. And again, with many of our collaborators, we've been mapping these disease genes that occur spontaneously in these new individuals that land on these synaptic proteins and cause dysfunction. I think this is important to understand because you will all know, of course, that people say that these disorders have a hereditary component, but we shouldn't underestimate the importance of new mutations. And there are many stories where apparently normal families have normal parents and even normal siblings and then affected individuals for many different sorts of reasons. And this technology is going to be important. I'll show you a few famous families. Here's the Kennedys. Joe and Rose Kennedy had many children, JFK being one of them. Rose Marie Kennedy was diagnosed as a young girl to have some mild intellectual disability, was thought to have schizophrenia. Her father organized for her to have a lobotomy, and she was then placed in an institution for another 50 years and largely forgotten and ignored by her family. Albert Einstein and his first wife had two sons, Hans, who was perfectly normal, but Edward, when he went to medical school after the age of 18, started to show the early signs of schizophrenia. He was institutionalized in Zurich and spent the rest of his life for another 50 years in an institution. And here are the parents of this. Here is the mother, Edwina Williams, mother of a daughter. This is Rose, and this is Thomas, and later known as Tennessee Williams, a famous playwright. And he wrote very affectionately of his sister, who had also developed schizophrenia and had a lobotomy and was institutionalized, and he wrote about her in The Glass Menagerie. And there is enormous suffering that goes on within these families. So what can we do about it? And I would say that what we need to be doing about this is save our synapses. And we really need to be doing something on this front on several different ways. One way is prevention. Let's see what we can think about with respect to prevention. And I'd like to just say something about drugs here. And here are various types of drugs, hallucinogens and even alcohol. And scientists have tested to what extent these drugs actually have anything to do with those proteins I've been telling you about in the synapse. And it turns out that if you mix those drugs effectively with mutations here, you can get very unusual and unexpected effects. And something that I think we'll see more of in years to come is this recognition that many different drugs of abuse that are in the community are having very deleterious effects on people who already carry mutations. In some other individuals who don't have those mutations, they'll probably just be fine. I think that's going to be a very important area of investigation. But if you have a family history of psychiatric illness, stay away from these. Head injury, stroke, and other forms of damage can impact on various sorts of synapses as well. And there's much work to be done on understanding that. But clearly, one of the important areas for the future is to develop new medicines. And although I've been talking to you about very fundamental aspects of the organization of molecules and the evolution of those genes and those forms of behavior. We have learned by doing these sorts of experiments that one can use these sorts of psychological testing paradigms to potentially diagnose and monitor the progression and onset of diseases, as well as find ways to test new drugs that might reverse the effects of mutations on these many different synaptic proteins. And that will be something for us in the future. So I'd like to sort of leave you now and just cast your eye across the story that I've just introduced you to, which is to explore the complexities of behavior and diseases and trace them back not just through their psychological components, but through the origins of the genes and the species that have arisen in the deep ancestry of humans, extending back hundreds and hundreds of millions of years, and the importance of these proteins and the molecular machines and the DNA that encodes those. And I hope you've found this lecture stimulating and enjoyable, but I cannot finish without extending an important acknowledgement to all of the many, many people who've worked in my own laboratory, as well as many individuals who've worked in collaboration with my laboratory, and I recognize many of you here in the audience. And there's many also in other countries and other parts of Europe. And it is within this program that we've had called the Genes to Cognition program, where we have assembled this research team and collaborators to address some of these questions that I've told you about. And we've been supported very generously by a variety of different funding agencies. I'd like to also thank the British Neuroscience Association for supporting this lecture. And for those of you who'd like to learn more about some of the science, we have an education website called G2C Online, and there's also another website called genestocognition.org, where we have lots of fundamental science resources. And with that, I thank you.